Good afternoon.
Good afternoon, sir.
Shall I start? Just a minute, just a minute. Are you able to see my screen? See my PPT? Yes. Okay, fine. So the last three classes, actually two classes together I took. The first class we discussed in general about insects, its relationship with its phylum, other members of the phylum Arthropoda. Then we discussed a bit about the history of entomology. Slowly we moved on to the dominance of the reasons for dominance, why insects are so dominant in our universe. Then we discussed about the first morphological part, the insect integument. We discussed various layers of the integument, what are the component, major component, that is chitin. Then we discussed about the appendages or cuticular outgrowths of insect body. Now there is a process called, the last class we just mentioned about it. There is a process called as molting in insects. This molting, as I told you, it's similar to the removal of skin by snakes. നമ്മൾ മലയാളത്തിൽ പാമ്പ് പടം പൊഴിക്കുന്നത് പോലെ എന്ന് പറയും. Many of you might have seen, at least not the process of removal of skin by the snakes, but at least the empty skin, the discarded skin of snakes you might have seen. So similar to that, the insects also remove periodically shed its old cuticle and forms a new cuticle, and this process is called as molting. I'm repeating, insect periodically sheds its cuticle. Just a minute, just a minute. Aparna. എന്തിനാ അങ്ങനെ ചോദിച്ചില്ല ഇടണം ആണോ ചോറിനോ ഓക്കെ അതൊന്നും വിഷയമല്ല തൊണ്ണൂറ്റി ആ ആ ആ ആ so sorry. So the insects shed their cuticle periodically, and the process of shedding this old cuticle is called as molting. So what is the need for this molting? Why insects molt? The major reason is, as we discussed in the last class, the insect integument or insect body wall is an extremely hardened sclerotized structure. So if you are inside an extremely hardened outer body wall, your chances of growing your body is extremely limited because your entire body is covered with a very hard structure. That hard structure, which is extremely sclerotized, it won't allow you to expand. It won't allow the body to grow. So what the insect will do? Insect will periodically shed its cuticle. That outer body wall it will remove. And in the last class you have studied, once the cuticle is formed, especially the procuticle, that is the most hardened part. In the procuticle, the hardening procuticle gets differentiated by the process called as sclerotization. Only the outer part of the procuticle, that is exocuticle, gets sclerotized or hardened. Endocuticle is less sclerotized. So by the time molting occurs, the old cuticle is shed, and before the formation of new cuticle and the sclerotization completes, before the completion of the sclerotization, insect has a small time interval to grow its body. So the insect grows its body along with its molting. Just after removing its old skin, it expands its body and then forms the new cuticle. Cuticle will be formed at that time, but the hardening process takes place a bit later, so that by the time the insect will have grown its size. Are you clear with molting? Yes. Okay. Now, the molting has two stages. The first one is apolysis and the second stage is ecdysis. I'm repeating, molting has two stages. First one is apolysis and the second one is ecdysis. Apolysis is the separation of the old cuticle from the new cuticle. Say, remember, imagine this is the old cuticle. So a new cuticle is slowly forming. So by the time the new cuticle is forming, the old cuticle slightly separates out from the new cuticle. So there will be a gap between the old and the new cuticle. And this separation of this old cuticle from the new cuticle is called as apolysis. And once this separation and once the new cuticle is formed, the old cuticle will be removed, shed. And this process of removal of old cuticle is called as exuectasis. The removal of the old cuticle is called as ectasis. So, molting has two stages. First one is the separation of old cuticle from the new cuticle, that is apolysis. And the second stage is the shedding of or the removal of the old cuticle, that is called as ectasis. And the removed skin or the molted skin, the old skin which is discarded by the insect is called as exuvia. Exuvia. Just like just a minute. That means the just like you have seen the discarded old skin of the snake, just like that you can also, if you observe closely in the environment, you can also see such discarded integument of the molted skin of the insect, and that is called as the exuvia. And once the ecdysis, that is the shedding of the old cuticle, is complete, the new cuticle will get formed. New cuticle will get formed. We will discuss about the process of molting process in detail, how the new cuticle is formed. Are you clear up to this? Shall I move to the next slide? Okay. See, this is the picture depicting the molting process. Forget about this ant which you are seeing over here. It has nothing to do with the molting process. It's just like a person, you are trying to capture a very good frame, and uh, sometimes you can see a person intruding in between that frame and spoiling that frame. So here ant is spoiling the frame. So remove ant from this equation. Ant has nothing to do with this ecdysis. So this is a grasshopper. This grasshopper is shedding its old cuticle. By that time the new cuticle is formed, you can see. And this old cuticle discarded, you can see it is just a skin, outer skin. And this discarded skin, if you see it, it has the perfect shape of the grasshopper. That's why we call it as an exoskeleton. It provides shape to the body. So it provides shape to the body and it is discarded. And this discarded or the molted skin is called as exuvia. Exuvia. Okay. This is the process. See the next stage by by which the grasshopper comes out of the exuvia. This is the third stage. It has almost completed, and now it has left. See, in all the frames you can see an ant. I told you to avoid the presence of the ant. Why this ant is doing? The ant, once this uh, once the molting process is completed, the grasshopper will excrete. The excreta contains some say sugary materials, and that kind of sugary materials is a feed for ants. That's why ant is not leaving the site of this process, molting process. So by this time the insect gets separated from the old cuticle. The old cuticle is removed, shed, and that is called as the exuvia, and you can see the new insect. So by this, at this, just after the ecdysis process, that is the shedding of the old cuticle, the new cuticle will not be sclerotized, that I told you. So at that time, before sclerotization, what the insect will do is, insect will take in a large volume of air into the body and it will expand. Insect will take in a large volume of air, and that volume of air will help in expanding the integument. That happens before the sclerotization process occurs. This is how insect grows. And one thing you have to remember, this process of molting and all occurs from in the young stage. In the adult stage, it does not happen. You are familiar with a butterfly. In butterfly lays the eggs. The egg hatches into a larvae. You might have seen a smaller larvae. How many of you have reared a larvae during your childhood? Anyone? Yes, Idea Lakshmi. She might know how it. If you provide the feed, it slowly grows. So what happens is that during the larval stage, say we usually say that in a normal lepidopteran insect, usually it have 5 stages. In each stage, after completing that stage, it molts and it grows and forms the new sclerotized cuticle. It feeds for 3 to 4 days. Then it by that time the body will be completely grown. There is won't be enough space. At that time, it again goes for a molting. The molting process helps in its growth. Then grows into a bigger larvae. Like that, till it pupates, it grows on molting, say 4 to 5 times. Each insect has a definite number of molting. You remember that each insect has a definite number of molting. It won't change with a particular insect. Usually for a butterfly, it is about 5 molting. And by the time the last, we call it as the last stage, last larval stage, that is the bigger larvae, then it grows into a pupae. From the pupae, we get the butterfly or the moth. The adult butterfly or moth will not undergo molting. That means once the insect attains adulthood, it does not grow. It is just like human beings. Many of you might be about 20 or 21 years old, isn't it? 19, 20, 21 may be a bit higher. Yes or no? Yes. So, are you growing in height now? Whether your height is increasing now? No. Nasreen is so unhappy that she is not growing in height. Okay, up to about maybe 16, 17, you might have grown in height. That means once you mature, the growth stops. That's the same thing happening in insects. Once the insect reaches the adult stage, there is no molting because there is no need for its growth further. The size remains the same. Only in the younger stage, where it has to grow in its size, molting occurs only in the younger stages. At that stage, it molts, and once the molting process is over, the shedding of the old cuticle is over, it has a short time to expand its body wall and it helps in its growth. So a small larvae, it will remain small for a few days, 1 or 2 days. After that, it undergoes molting and it increases in size. At that time, it feeds on for another 3 to 4 days. After 3 to 4 days, again, it needs to grow in size. So again, it undergoes a molt. It again and increases in body size after undergoing a molting. Starts feeding again. During the molting also, it won't feed. It grows again. It feeds on the leaves again, and it again grows. At that time, it again undergoes a molting. So this is how the insect grows. I will share a YouTube video. You can see it how it happens. This is a stage by stage process wherein you can see how a molting occurs. See, slowly, the grasshopper is shedding its old cuticle and it is coming out of the old cuticle, and the old cuticle is discarded. Now, what are the steps in the molting? I told you, first step is apolysis, that is separation of old cuticle from the new cuticle. And once the old cuticle is separated from the new cuticle, the space between the old cuticle and the new cuticle, actually what it happens is it's not new cuticle will not be formed. First, the old cuticle will be separated, and cuticle just below the cuticle it is epidermis. You remember the last class. It consists of three layers: cuticle, the hypodermis, the basement membrane, hypodermis, and the cuticle. So what happens is during apolysis, first the old cuticle will get slightly separated from the epidermis, and there will be a space between the epidermis and the cuticle. And this space is called as the exuvial space. During the apolysis process, the old cuticle will get separated from the epidermis, and that space created is called as the exuvial space. And into that exuvial space, the space created between the epidermis and the cuticle, a fluid is secreted by the epidermis. That fluid secreted by the epidermis is called as the molting fluid. The fluid secreted into the exuvial space by the epidermis is called as the molting fluid. And this molting fluid contains number of enzymes such as chitinases and proteinases. As the name indicates, chitinases are those enzymes which has the ability to degrade chitin. Proteinases are the enzymes which has the ability to degrade proteins. So this chitinases and protein, you might be remembering in the last class, cuticle consists of mainly of chitin, about 20 to 50% of dry weight is chitin, and it links with the proteins, sclerotin, resilin, etc. So the major components are chitin and protein, and the molting fluid contains these enzymes, chitinases and proteinases. But just after the molting fluid is secreted into the exuvial space, these enzymes will not be active because this proteinases itself can harm the epidermis it's secreted. The epidermal wall, you might have studied about the body wall, the cell wall of cell membrane of each organism. The cell membrane is made up of lipids and proteins. Lipid protein bilayer, you might have studied fluid mosaic model and all. So if there is a proteinase present in the fluid, that proteinases can harm the epidermal wall itself. So just after the secretion, these chitinases and proteinases will not be active at the time of secretion, so that it won't harm the epidermis. After, just after the molting fluid is secreted, the insect, the epidermis forms the first layer of the new cuticle, and that is the cuticulin layer or the outer epicuticle. You might be remember inner endocuticle and outer epicuticle and inner epicuticle. You might be remembering the epicuticle will have four layers: cement layer, wax layer, outer epicuticle, and inner epicuticle. Outer epicuticle is also called as the cuticulin layer. So once the space is created, exuvial space is created, molting fluid is secreted, it contains enzyme, that enzymes won't be activated at that time. Then the epidermis forms the first layer of new cuticle, that is the cuticulin layer or outer epicuticle. And once the cuticulin layer is formed, the enzyme gets activated, so that the enzyme will not act directly on the epidermal cell wall and epidermis will not be harmed. So cuticulin layer, once the new cuticulin layer is formed just above the epidermis, then only chitinases and proteinases becomes active. So if you are asked which is the first layer of cuticle formed during the molting, it is the cuticulin layer or the outer epicuticle. And then The enzymes, once it gets activated, the chitinases and proteinases digest the endocuticle. The procuticle is divided into exocuticle and endocuticle. Only the endocuticle will get digested by chitinases and proteinases because the exocuticle is heavily sclerotized, and even this chitinases and proteinases cannot digest the exocuticle. Only the endocuticle will get digested, and this molting fluid containing the digested chitinases and proteinases will be resorbed or absorbed back by the epidermis. This will help in conserving the resources for the insect because it can be utilized again for the secretion of new integument. So once the chitinases and proteinases digest the endocuticle, this endocuticle will be digested endocuticle along with the molting fluid will be resorbed by the epidermis. Are you clear up to this? Audio is breaking. Someone was telling my audio is breaking. Is it so? No. Okay, fine. So immediately after the formation of the just a minute. Okay, I will a few sentences. It is one slide. I think I have missed in between somehow. So this is the molting process. This is the cuticle. You can see the epicuticle, the procuticle, which gets differentiated into exocuticle, endocuticle. This is the hypodermis or epidermis, and we have the basal membrane over here. And during the apolysis process, there is a space created, and that space created is called as the exuvial space. And into that exuvial space, the molting fluid is secreted by the epidermis, and this molting fluid at that time it is inactive, and the new epicuticle, that is the cuticulin layer, is formed over here. You can see the dark color, and once the new epicuticle is secreted, it forms the molting fluid. The enzymes will get active and it completely digests the endocuticle, and the exocuticle will not be digested. And this digested endocuticle along with the molting fluid will be absorbed back. By that time, new procuticle will be formed. New procuticle will be formed. So while at that time the the new procuticle which is formed is undifferentiated, that means it is not differentiated into exocuticle and endocuticle, because at that time sclerotization will not have happened, and the old cuticle is completely devoid of the endocuticle, and only the exocuticle and the epicuticle will be remaining. That will be discarded and the new cuticle will be formed. Just a minute, I will sorry. Okay, here it's that sentence. So once the endocuticle is completely digested and the molting fluid is resorbed, as I told you, the insect will take in a huge quantum of air to exert pressure on the new cuticle, which is not sclerotized. Procuticle is formed, but it is not sclerotized, undifferentiated at that time. So exerting pressure on the integument will help in its growth. And in its growth also, it also helps in the shedding of the old cuticle, that is the ecdysis process. In certain insects such as dipterans, flies, housefly and all, the head has a reversible sac. That means just like a balloon. Just imagine that the fly head has a balloon over here. So what it will happen is that once it sucks in air at the time of molting, this balloon will grow out. It is, we call it as an reversible sac, and that balloon will grow. Because of this growth of this balloon, the uh, um, cuticle, old cuticle will get broken and it can be easily removed. I will take the example from a medical field. You might have heard about angioplasty. What do we do in angioplasty? If you have a block in your coronary artery or coronary vein, anyone? No? We will. You might have heard about putting a stent into the artery or veins. So, in order to put that stent or vein, there will be a block in the artery or the vein. Nerves from the that is bypass. Bypass nerves on the leg is taken. That is bypass. I'm not talking about bypass. I'm talking about angioplasty. Angioplasty, if your coronary artery or any artery or vein in your heart is blocked, then you will insert a probe into that area, the artery or vein, and at the end of the probe there will be a balloon, and once the probe reaches the yes now Nifa is telling, once the probe reaches the blocked area, a balloon kind of thing will be expanded so that the block will be removed, so that the artery also, once the balloon is expanded, the artery also expand the same manner. Diptera has a balloon-like reversible sac at the head, and once during the molting process, when the insect takes in the in, this reversible sac protrudes and it breaks the old cuticle and helps in the molting process. And that structure in the head of dipterans is called as telenum. You can see it over here, telenum. Telenum. That is an reversible sac at the head of the flies. Others normal way, because of the pressure exerted by the air sucked in, it breaks. And as I told you, one procuticle will be formed. It is not sclerotized, it's expanded. Once the expansion, the growth is completed, then the procuticle will get differentiated into exocuticle and endocuticle by the process of sclerotization. Then wax layer is secreted over the outer epicuticle or cuticulin layer, and at last the semen layer is secreted. So the first layer to be secreted in the new cuticle is the outer epicuticle or cuticulin layer, then forms the procuticle, then forms the wax layer, and at the end semen layer will be secreted. Okay, this is the process. This is again a new picture. See this. Here you can understand how the insect is not sclerotized. See this, once it is sclerotized, I told you in the last class, sclerotization means darkening or darkening and hardening of the cuticle. See the old cuticle, the exuvia. It is dark brown in color. See the newly molted insect just after coming out of the old skin. It is white in color because the procuticle is not sclerotized. It is not hardened. At that time, it sucks in air and it can grow. Then the sclerotization happens, and then then onwards it can won't be able to grow. This is again another picture wherein just after molting, it is whitish in color. Slowly the sclerotization occurs. Time is less. Whether you want to watch a YouTube video of this molting process, or should I share the link? Okay, just a minute. Just a minute. Can you see my video? Yes. This portion I have not told. You will study about it. Remember here they're selling cuticle layer and polyphenol layer. Actually, it is different. Scientists used in different terminology. So polyphenol layer is the outer epicuticle, and what they told us the cuticle layer is the inner epicuticle. Okay.
Sir, you are muted.
Could you understand how what is the structure of integument looks like? Yes. Just a minute. Let me. I will try to show a molting process. Okay. This is the cicada I told, which has 17-year life cycle. See, it's slowly expanding, shedding its old cuticle. See, it is taking up air. You can see that it is taking up air. The old cuticle is slowly getting broken. It is emerging out of the old cuticle. Yes. See the wings. See, now the wings are formed. It will expand now. See, see the size now. It is bigger than the old one. See this slowly, slowly the color changes, slowly gets sclerotized. So you can see a lot of this kind of videos. Hope you are familiar with molting process. Any clarifications? Okay, fine. Just give a random search in YouTube. You can find a lot of information. You can understand also better because with the help of animations. Okay. Okay. Because of the shortage of time, I'm not stopping over here. I'm moving into the next topic because it's a huge course. I don't have enough time to give you some spare time. So sorry for continuing the class. Uh, I'll just give a brief introduction about uh, insect head. Okay, so I will share it. So in the last two classes we have seen about the integument, that is the outer body wall. Now we are going to see each body part one by one. As you might be remembering, the insect body is divided into three regions: head, thorax, and abdomen, and that division is called as tagmosis. Each region is called as tagmata. First, we will be taking up head. We will be dealing in detail about the head, what are the structures on it, and at least 2 or 3 classes we may need for head itself, head alone. So, sharing the PPT. Okay. So, first let's see how the insects are classified based on the type of head. There are certain insects which are called as hypognathous, some are prognathous, certain others are opistognathous. I'm repeating, based on the position of mouthparts on the insect head, the insects are classified into hypognathous, prognathous, and opistognathous. Hypognathous, prognathous, and opistognathous. If the mouthparts are directed downwards, then that kind of head is called as hypognathous. I will show you. Don't feel that I'm just reading out. I will just first describe it, then I will show you how it looks like. So, if the mouth parts are directed downwards, then that kind of an insect head is called as hypognathous. If the mouth parts are directed forward or anteriorly, then it is called as prognathous. If the mouth parts are directed backwards, then it is called as opistognathous. So, if the mouth parts are directed perpendicularly downwards, it will be like this. Then it is called as hypognathous. If the mouth parts are directed forward, it is called as prognathous. If the mouth parts are directed backwards, like this, it is opistognathous. So, if it is like this, it is perpendicular. If the mouth parts are perpendicular, then it is hypognathous. If it is directed forward or anteriorly, it is prognathous. If it is directed backward like this, then it is called as opistognathous. So how I will just give a small way to easily understand it. Just a minute. See, I am drawing a small depiction of it. Consider this as the head. This is the thorax, and this is the abdomen. This is the normal kind of thing. So in a hypognathous head, this is the thorax, this is the abdomen, and the head will be directed downwards. The mouth parts will be remaining. Oh, just a minute. The mouth parts will be remaining over here in this area at the tip of the head. So the mouth part will be directed downwards. And in the case of prognathous, it is the same as first I drew, typical kind of thing. Head, thorax, then abdomen. The mouth parts will be directed forward. Here it will be the mouth part. And in the case of opistognathous, this will be the thorax, this will be the abdomen, and the mouth parts will be directed backwards. That is opistognathous. Are you clear? Hypognathous, prognathous, and opistognathous? Yes. Now, in entomology, you should remember about examples. Whenever you are writing an exam with respect to this terminology, always remember their examples. Hypognathus, typical example is grasshopper. I will show the pictures, then you will be able to understand better. Prognathus, it is beetles and ants. We can catch hold of an ant and observe how the mouth parts are directed. Then Opistognathus, it is bugs. Hypognathus example is grasshopper. Prognathus is beetles and ants. Opistognathus, it is bugs. I will show the pictures. See this. See this grasshopper. This is the head, thorax, abdomen. See the mouth parts. This is the mouth part. Actually, this is the mouth parts. These mouth parts are directed downwards. This is the prognathus head. See the mouth parts directed forward or anteriorly. These are the mouth parts. In Opistognathus, you can see this is the mouth part. These mouth parts are directed backward. Yes, now you can see it. This is the hypognathus head. The mouth part over here. This is a prognathus one. This is the mouth parts directed forward, directed anteriorly. This is the opistognathus one. This is the mouth part which is directed backward. Okay, fine. Okay. So, what is the structure of an insect head? I will always take the example of, I will try to explain the insect head based on our own head itself, so that you will be able to visualize it. Certain things you may not be able to see it properly. Certain things you can see it only under microscope. So I will always take the example of head to explain this insect head, different parts of insect head. So the head is a sclerotized, heavily sclerotized capsule with a number of grooves or sulci. Sulci is the plural, sulcus is the singular. So just like our head, skull, it is extremely hardened. So like that, it is just like a capsule. It is just like very hardened sclerotized capsule. But before in the young stage, there is no sulcus and all. But some persons you might have seen നെറ്റിയിലെ ചുളികൾക്ക് എന്താണോ ഇംഗ്ലീഷിൽ പറയുന്നത്? You might have seen in, if you hold it like this, you can see it some grooves over here. നമ്മൾ നെറ്റ് ചുളിക്കുക എന്ന് പറയില്ലേ? Not scales. It's not scales. Wrinkles. It can be called as wrinkles, but it's not exactly wrinkles. Once you age, if you have some aged grandfather or grandmother in your home, you can see that numerous wrinkles, sulcus, just like sulcus. That is invaginations of the cuticle. In insects, that invagination it is present on the head all throughout its life, not only in the age, in the older stage. That is because that invaginations provides rigidity or strength to the sclerosis, the cuticle of the head. Yesterday last class I told you with respect to the sheet and all, that corrugated sheets and all. So that invagination or sulci or grooves on the head provides strength or rigidity to the insect cuticle. Can you answer how many segments are there in an insect head? Last class I told you. How many insects are there? How many segments are there in an insect head? 6. Good. So, though there are 6 segments, you cannot see the segmentation in insect head because all the segments are fused together into a single sclerotized capsule, and that segmentation in the insect head is not visible. And the cuticle, I told you, it is inflected to give rigidity, and because of the presence of these grooves, so imagine if you have a groove like this, say if you have a groove like this in a circle, then you can say that your head can be divided into number of areas. the area inside the groove. So likewise, the insect head, because of the presence of numerous sulci, it can be divided into number of grooves and number of areas. I will give another example. You might have seen uh, yeah, boundary walls you have seen. You separate out your area based on boundary walls. In your every home will have a boundary wall, and that separates out the our area from the neighbor's area. So imagine that instead of this boundary wall, we have a furrow over there. ഒരു ചാല് കീറിയേക്കാണെന്ന് വെച്ചു. Instead of a boundary wall, you dig out a furrow over there, and that furrow helps in separating out the area. Similarly, this sulcus present on the insect head helps to divide the insect head into number of regions or sclerites. And this head is sclerotized. I told you the segments fuse together to form get completely sclerotized. It is sclerotized in the dorsal area, the top part. The lateral wall is sclerotized. The anterior wall is sclerotized. The posterior wall is sclerotized, but the ventral wall is not sclerotized. So, dorsal, lateral, anterior, and posterior walls of the insect head is sclerotized, while the ventral wall is not at all sclerotized, because at this ventral area we have the mouth parts. It's not sclerotized. Up to this, is it clear? I'm stopping. Okay. If you feel that I need to change the method of teaching, you can easily inform me. I think almost all of you might have my WhatsApp number. Just give a message if you find it difficult to understand these things. Because from now onwards, the things may get slightly difficult. You have to imagine certain things, because certain things I may not be able to show you properly. So whenever if you face any difficulty or if you have any doubt, and if you are hesitant to ask in the class, you can just give me a message in the WhatsApp. I will reply surely, whatever be the question you face. Okay, anything? Today Vishnu is not there. Your lady rep morning itself gave me a message. Lakshmi, she's the one who messaged me. Yes. Okay, fine. Thank you.
Thank you, sir.
Okay.